Hello everyone, I am Dinesh Urpi. In this video, we'll continue with the second session and we will study the restriction enzymes, ligases and nucleic acid modifying enzymes which are used in recombinant DNA technology. Restriction enzymes. As we already mentioned in another video, the gene manipulation technique or the recombinant DNA technology involves many type of chemicals and many enzymes. Some of the important enzymes like restriction enzymes, ligases, etc. So the first we are going to study is restriction enzyme. They are also commonly called endonucleases because they cut the DNA inside, so I mean within, not at the end. So in the middle of the DNA, because they cut the DNA at middle of the enzyme, middle of the molecule, they are commonly called endonucleases. And because they cut the DNA at a restriction site, they are commonly called restriction enzymes. So what are these restriction enzymes? Restriction enzymes are the enzymes which are found in bacteria. So you find these enzymes naturally in the bacterial cytoplasm. They are commonly called molecular scissors because they are known to cut the DNA molecules. Since they cut the DNA molecule and DNA is a molecule, so they are commonly called molecular scissors. They cleave DNA molecule at restriction sites. So this is very important. These DNA cutting enzymes are very specific. So they cut DNA molecule not randomly. They cut the DNA at a restriction site. So every restriction enzyme has its own restriction site. So it will go and cut the DNA at that site only. For example, so you consider the enzyme, one restriction enzyme, which is a very common, which is the most common enzyme, that is EcoR1. And this is the restriction site, right? So you can see here, this is a sequence, this is the part of DNA where this EcoR1 is going to cut the DNA molecule. So this is the restriction site showing a particular sequence, right? So that's why they are called restriction enzymes. It will not cut the DNA anywhere else. If it finds this particular sequence, there only it's going to cut. So that is the significance, that is the importance of these enzymes. And there are more than 3000 different types of restriction enzymes identified in different bacteria, of which around 200 are commercially available for gene manipulation techniques. So in the genetic engineering, many enzymes are used depending upon the requirement. So the suitability and requirement, the genetic engineering techniques utilize different type of restriction enzymes. And restriction sites are normally palindromic sequence. Now this is what you have to understand. As I already told you, this enzyme is going to cut the DNA molecule at a particular site, and that site is called restriction site. Now what is a restriction site? So restriction sites are normally they show a character called palindromic sequence. So here palindromic sequence means they read same in opposite direction. You consider this. So this is the restriction site of EcoR1. So EcoR1 is the restriction enzyme which is extracted from Escherichia coli bacteria, and this is the first restriction enzyme that is extracted and utilized. See, read the sequence here. CT, okay, I'll start from here. GAATTC. So this is the sequence. Now in the opposite side, you can see from opposite side, it reads the same. It is GAATTC here and it is GAATTC. So such sequences which read same in both directions, such sequences are called palindromic sequences. So the restriction sites for the restriction enzymes, they are palindromic sequences. And when they cut the DNA molecule, they are going to produce the sticky ends. That means they will not produce blunt ends, right? So the restriction enzymes are going to produce the sticky ends, means this is a single stranded DNA which is produced due to the cleavage or due to the cutting. And this part, so this is one DNA molecule, this is another DNA molecule. So this is the DNA from one organism and this is DNA from another organism. And here, same type of enzyme, if it is used to cut the DNA, both the DNA are going to be cut with the same restriction enzymes, right? That means both the DNA have same restriction sites. So it is possible to cut both the DNA with the same enzyme if they have the restriction sites, if both have the same restriction sites. And here, this DNA, when it is cleaved, it is going to produce a sticky end like this. And this DNA is also going to produce a sticky end. Now, what is the sticky end? Sticky ends are complementary to each other. So this sequence and this sequence is complementary. So when they come nearer, the hydrogen bond itself will help in pairing, right? Of course, to join both of them, we need another enzyme called ligase. So that is the specialty of restriction enzymes. So they are called molecular scissors. They are extracted from bacteria and they cut the DNA molecules at a specific site called restriction sites. And once they cut the DNA, they are going to produce sticky ends, which is a very important aspect in the genetic engineering. What are endonucleases? What are restriction enzymes? Of course, these are the enzymes which are utilized in the genetic engineering. Now the question is, why they are present in bacterial cytoplasm? Let us try to understand it. You know, in higher organisms like humans, we have something called immune system. So whenever we get infected, say if I get infected by a virus or a bacteria or a protozoan, I have a system called immune system, and my immune system will produce antibodies or it will produce different type of cells which will destroy the pathogenic organism which has entered into my body. So this is called immune response. So I have an immune system which protects my body against the infection of pathogens. Now bacteria, say this is a bacteria, let us say this is Escherichia coli bacteria. Bacteria is a very simple cell. It is a prokaryotic cell even without a nucleus. So even the bacteria are infected by some viruses called phage viruses. They are commonly called bacteriophages. So as we get infected by bacteria and other pathogens, even the bacteria are also infected by these phage viruses. So how it is going to protect itself against the infection of this phage? So endonucleases can be considered as the immune system of bacteria. Say this phage, it will introduce its own DNA into the bacterial cell, which will multiply and which will ultimately destroy the bacterial cell. So when the DNA of virus is introduced into the bacterial cytoplasm, the restriction endonuclease enzyme of the bacteria is going to destroy the DNA of the virus, right? So as the bacteria which enters into my body, it's going to be destroyed by the antibodies of my immune system. In the same way, the restriction endonuclease enzyme of the bacteria will destroy the DNA of the virus. Now remember, the restriction endonuclease which is present in this bacteria, it will not cut its own DNA. The main reason is this restriction endonuclease will cut the DNA at restriction site, and that sequence is not found in its own DNA. In this way, its own DNA is protected from the action of this whereas this enzyme will protect it from the infection of virus. So that is the reason for the presence of restriction endonucleases in the bacteria, and discovery of these restriction endonucleases has changed the biotechnology or gene manipulating field. Here, let us try to understand how it works. So, creating a recombinant DNA with restriction enzyme. So, this is a DNA of one organism, and this is DNA of another organism. We can consider this as a vector, and this is the DNA from the source, or the gene of interest is present in this particular DNA. This is the restriction site, right? GAATTC. So as I already mentioned, this is a restriction site for EcoR1 enzyme. And using EcoR1 enzyme, when you cut this DNA, it will produce two fragments. You can see here, this is where it is going to cut it, and it has produced the sticky ends, right? So this is one sticky end, this is another sticky end. And because I'm using the same EcoR1 enzyme, because I'm using the same restriction endonuclease enzyme, the same restriction site will be the target, and it will produce the same type of sticky ends in another DNA also. Now I have two different DNA molecules with complementary pairings, complementary nucleotides. So that is why they are called sticky ends. And because of these sticky ends, now I can fuse this segment with this one, and this segment with this one. Like that, right? And here you can see here this segment, right? It is going to be fused with this particular part. Because the pairing is complementary, here it is AATTC and here it is AATT, so AT. So there will be a pairing, and the hydrogen bond formation will help in the pairing. And in the next step, these DNA fragments have to be fused. So it is called ligation, and ligase enzyme is going to uh, full do that work, right? So here, with the use of ligase enzyme, now I have a DNA fragment which is part of both the DNA. So this is actually a mixture of two different DNAs, which is a combination of two different DNA. Therefore, we call it as recombinant DNA molecule. Types of restriction endonucleases. As I already told you, it is a repetition. Restriction endonuclease enzymes are obtained from the bacteria. There are so many different type of bacteria available, and based on the source of bacteria, the restriction endonucleases are named. Remember, different endonucleases from different bacteria, they have different restriction sites. So they will cut the DNA at a particular restriction site. So based on the requirement, based on the restriction sites, the restriction enzyme has to be chosen. Now basically, restriction endonucleases are categorized into type 1, type 2 and type 3. Now in case of type 1, they cleave only one strand of DNA and they require Mg, magnesium ions, and ATP for its functioning. For example, EcoK, EcoB, etc. So here, this is the main thing. They cleave only one strand of DNA. Whereas type 2 enzymes, these are used in genetic engineering. So the restriction endonuclease enzymes which are used in genetic engineering, they are mostly type 2 enzymes, and they cut DNA on both the strands at restriction sites, and they do not require ATP for functioning. This is very important. And as I already told you, more than 3,000 are identified, and more than 200 are available in the market, like HINF1, EcoR1, PVU to ALU1, HAE. So here the name basically refers to the bacteria from which it is isolated, like HAE, it is obtained from Haemophilus bacteria, like that. Type 1, which recognize specific DNA sequence but make their cut at seemingly random sites that can be as far as 1000 base pairs away from the recognition site. So this is not useful in genetic engineering, because type 1 enzymes will cut the DNA away from the recognition site, whereas type 2 is very special because it will cut the DNA directly at the recognition site. Because to create a recombinant DNA, I need sticky ends. So that is achieved by type 2. Whereas type 3 endonuclease enzymes, they also recognize the specific sequence but make their cut at a different specific location. So because the type 1 and type 3 enzymes, they cut DNA away from the restriction sites, they are not useful in the recombinant DNA technology. Whereas type 2 restriction enzymes, since they cut exactly in the recognition site of the DNA, they are very useful in recombinant DNA technology. So the next enzyme which is extensively used in recombinant DNA technology is DNA ligase. The enzymes used to join fragments of DNA together. They are commonly called molecular glues, as restriction enzymes are called molecular scissors because they cut, and here they are called molecular glues because they join the DNA. Ligases join fragments of DNA together by catalyzing the formation of bonds between neighboring nucleotides. So the DNA is made up of nucleotides, right? So when two sticky ends come together, the pairing will occur because of the uh, complementary nucleotides, whereas the DNA strands must be joined together covalently. So the phosphodiester bond must be formed between the nucleotide of different DNA strands. So T4 DNA ligase. So the ligase enzyme which is obtained from T4 bacteria, that is one of the most commonly used ligase enzyme in the recombinant DNA technology. So here you can see, just to show you, this is one DNA, this is another DNA, and these are the two sticky ends which are produced by the action of restriction endonuclease cleaving. And this is the ligase enzyme. So ligase enzyme will help in formation of the phosphodiester bonds or the covalent bonds, a strong covalent bond in these two parts. So these two strands will be joined together, these two will be joined together, and now we have a continuous strong DNA molecule which is a recombinant DNA because it is formed by the fusion of two different DNA molecules. The majority of ligation reactions involve DNA fragments that have been generated by restriction enzyme digestion. Most restriction enzymes digest DNA asymmetrically across their recognition sequence, which results in a single strand overhang on the digested end of the DNA fragment. So that's what I was telling in the previous slide. The restriction enzyme, when it cuts the DNA, it produces the single stranded overhang. So this is the single stranded overhang and this is another single stranded overhang. And as it is cut with the same endonuclease enzyme, so they show the pairing, the base pairing, and that makes the job of joining them very easy because they will be paired, and at this point the ligase enzyme is going to join, it is going to create a covalent bond. And of course, as the restriction site is AATTC, GAATTC, this is a site which is generated by the cleaving of EcoR1 enzyme. So restriction endonuclease and ligase are two important enzymes which are used in recombinant DNA technology. There are many enzymes which are used which are essential in the gene manipulation technique. So let us try to understand some of them. We already understood restriction enzymes and how they are used, and DNA ligase. So the most important enzymes used in recombinant DNA technology. This is used to cut the DNA and this is used to join the DNA. Then polymerase chain reaction enzymes. Polymerase chain reaction is a technique where we are going to amplify the DNA sequence. So a small DNA sequence or a small gene which is extracted, which is isolated, you can create multiple copies of that gene or DNA using a technique called polymerase chain reaction, and it uses many enzymes, right? So DNA polymerase enzyme is the most important enzyme because you are going to create the multiple copies of the DNA. So DNA polymerase is used in the PCR, and new strands based on the template strand. Taq polymerase. So this is a polymerase enzyme which is mainly used in PCR technique. Then we have reverse transcriptase. Now remember, DNA polymerase enzymes are naturally found in the DNA in the cell, and they are used to create copies of DNA. You know, DNA multiplication or DNA duplication occurs during cell division, right? So basically, every cell which is capable of dividing, it contains DNA polymerase. But for PCR technique, we need a special type of DNA polymerase, that is Taq polymerase, which is extracted. Reverse transcriptase, as the name suggests, these are the enzymes which are capable of producing DNA from RNA. You know, transcription is the process of formation of mRNA from DNA, and transcriptase is the enzyme which will do it. So during protein synthesis, mRNA is produced from DNA template. But uh, this is a technique. So for example, when we are creating DNA libraries, right? So this is a reverse technique where we have mRNA, right? So you know mRNA is responsible for the production of a protein. So that means we have an mRNA, we know what protein it is going to produce. So the gene product we already know. And I want to understand, I want to know the DNA sequence. So I have to go in reverse. So in this technique, we use mRNA, and from the mRNA, DNA is produced. So for that purpose, reverse transcriptase enzyme is used. And phosphatases. These enzymes are used to remove phosphate group from DNA molecules. They are often used to prevent self-ligation of vector DNA molecules during cloning. So we extract we extract vectors from different sources of bacteria. And when the endonuclease enzyme is going to cut the DNA, there are high chances of that cut DNA joining or itself. So it's called self-ligation. So since our intention is to introduce the gene of interest in that vector, so I don't want the self-ligation to happen. So to prevent that, phosphatases are used. So it will remove the phosphate groups from the DNA molecule. Next, most commonly used enzymes are kinases. Kinases are enzymes that are used to add phosphate groups to the DNA molecules. They are often used to label DNA fragments with radioactive or fluorescent tags. So once the recombinant DNA is introduced into the target cells, I want to screen it. And one of the technique of screening is use of these fluorescent tags. So kinases are the enzymes which can be used as a fluorescent tags. Then exonucleases. These are the enzymes which are used to remove nucleotides from the ends of DNA strand. They are often used to generate blunt ends of DNA fragment. Exonucleases cut the DNA from the end and they create blunt ends, right? Whereas endonucleases cut the DNA at the in between. and they create sticky ends. So that is the difference between endonuclease and exonuclease. Exonucleases are also used for gene manipulation technique or recombinant DNA technology. So remember, all these enzymes are naturally available. They are there in the nature from the beginning, and we have extracted them and we did some modifications as per our needs, and they are used in gene manipulation techniques. So in this video we studied some of the important enzymes which are used in recombinant DNA technology. So again, here are the references, and there are many other sources which you can refer for detailed study. So thank you.